Hello and welcome to the discussion on membrane transport proteins, and this time the lecture is on aquaporins. Now aquaporins are water channel transport proteins on the plasma membrane, and as you can see, this is the plasma membrane with the hydrophilic head, hydrophobic the core, extrinsic proteins, intrinsic protein, and some transmembrane proteins are also shown here. And aquaporins actually form this type of channel proteins, transmembrane proteins, which lie along the length of the plasma membrane. Now, water actually, because of its high concentration outside or extracellular fluid, they do cross the hydrophobic core of the plasma membrane by diffusion. But diffusion is a very slow process for the entry of water into intracellular fluid. So there is the significance of aquaporins. And as often in the case of science, the secret of water channel, aquaporin, was discovered by chance in 1992 only, while working on erythrocyte membrane proteins. And Peter Ego was awarded Nobel Prize for this discovery in 2003. Now, aquaporins actually belongs to the transport by aquaporins belong to the facilitated diffusion here. So it's obviously comes under passive transport, that is downhill transport, and among facilitated diffusion, this is coming under transportation through protein channels. Now, aquaporins are definitely, they are not the usual ion channels, but they are integral transmembrane proteins which exclusively transport water alone. And the transport is obviously based on osmotic gradient, that is from extracellular space where there is a high concentration of water, that water actually osmotically gets into intracellular fluid with the help of aquaporins. And these molecules, aquaporins, these protein molecules are especially abundant in RBC membrane and in the membrane of cells, those cells which secrete fluids, for instance like tears on tear gland, saliva and salivary gland, as well as in kidney cells, which cause reabsorption of water from primary urine. And transport of water by simple diffusion, as I said earlier, is a very slow process, but with aquaporin, through aquaporins, it is very fast, as fast or as high as 1 million molecules per second. So that is pretty fast. Now let us go through the structure of aquaporins. Aquaporins actually, they form 4-part clusters or tetramers in the cell membrane, embedded in the cell membrane, with each of the 4 monomers acting separately as water channels. So they form separate water channels. And when we take each of the monomer, the structure is like this. Each of the monomer is actually composed of a bundle of 6 integral transmembrane α-helices, which means that each of these monomer is having 6 α-helices embedded in the membrane, and they are actually embedded in the membrane with the amino group and carboxyl group facing the inside of the cell. Further, the monomer actually forms a channel or cavity, and there are hydrophilic amino acid residues which line the cavity of this channel to provide a polar environment to interact with water which comes through it. And at the same time, there are hydrophobic amino acids like this. Hydrophobic amino acids which are placed along the outer periphery of the molecule, which will interact with the hydrophobic core of the plasma membrane and stabilize the molecule. And another feature of aquaporin is the presence of positively charged amino acid residues which are placed along the path of the channel, which repel the cationic molecules or protons, H+ ions, from sneaking through the cavity. As we know, unauthorized entry of cations or protons can cause imbalance in ionic concentration across the membrane. So it is actually to avoid such a situation that positively charged amino acid residues are positioned here. And this is actually done so effectively in that the entry of even hydronium ions are prevented.